hear me okay, can you?
Hi, thanks for having me. You can hear me okay, can you? Very good. Joachim, can I just say, I can't see the chat window, so if anybody puts in a question, do you mind just reading it out?
No problem.
Okay, thanks. Good, so hi, I'm Sam. Thanks very much for inviting me to this. Yes, so I'm going to be talking about kind of similar work to what other people have done, but looking very specifically at early life. So we know that over time, particularly during the early parts of our life, we're very dependent on what we call co-regulation when it comes to both emotion regulation and to cognitive regulation, across the parent-child dyad. And we know that over time we transition to a state where we're relatively more able to regulate on our own. So what I'm really looking at, and my group is looking at in our research, is what role does parent-child synchrony play in this transition, particularly from the point of view of neural synchrony? There's been some very brilliant work done by people like Ruth Feldman and many others looking at similar ideas in terms of physiology and behavior, but we're really concentrating on neural synchrony. So one particularly important question that I'm going to be talking about is interpersonal neural synchrony learnt, or is it there from birth? So just to present a summary of my talk, so part one, I'm going to be presenting a study showing the aspects of interpersonal neurosynchrony develop even by 8 months. And then I'm going to be talking about some research that we're doing on an ongoing basis, looking at what causes interpersonal neurosynchrony. And particularly, we're going to be distinguishing between two very different causes of synchrony. One, looking at entrainment to common behavioral cues during an interaction. And another looking at entrainment potentially in the absence of any behavioral entrainment as a function of shared understanding. So this is picking up on many of the themes that Simone has been talking about, and also that Alejandro is going to be talking about in the next talk. So yes, so just to present study one first. So this is a paper that was published a couple of years ago in Proceedings of the National Academy of the Sciences. This was done in collaboration with some colleagues in Cambridge and funded by the Economic Social Research Council of the UK. So what we did for this study was we did two experiments. Experiment two, we simply got an adult to interact live with an infant. The adult was reciting nursery rhymes. And we asked them to do it in two conditions. One, direct face-to-face gaze, and the other with the adult looking 20 degrees aside from the infant. So that we called indirect gaze. And we measured the EEG activity in both partners at the same time. And then in experiment one, which actually came first, we did the same thing, but we looked at the relationship between the live infant as they recorded a pre-recorded adult, and we pre-recorded their EEG. The reason that we did this is because with the pre-recordings, we could very tightly control for things like the volume and the pitch and the speed and the intensity of the speech in a way that we couldn't control as precisely in the live interaction. So we measured just from 2 electrodes in the infant and 2 in the adult, C3 and C4, either side of the vertex. And we just looked in this study at theta and alpha band fluctuations.
Sorry, Sam, could
sure?
you please show the slides full screen? Now it's in the presenter view.
Oh, okay. Yes, I think, let me just stop the sharing and turn it on again. And I think if I share my other screen. So we calculated, so yes, I was saying we looked just at C3 and C4 in theta and alpha band. And we calculated a measure called generalized partial directed coherence, which is derived from Granger causality. And it basically, like any assessment of Granger causality, it looks at changes, it looks at whether changes in one time series predict changes in another, in a, approximately put way. And we looked both at, do the child predict the parent, and does the parent predict the child? So in experiment 2, we found that during both direct and indirect gaze, we got Granger predictive relationships from the adult to the infant, yeah, and also from the infant to the adult. So the blue lines are showing infant to adult, and the red lines are showing adult to infant. These were, and when we compared them, these were stronger. Both the adult to infant and the infant to adult relationships were stronger during direct gaze compared to indirect gaze, although they were present in both situations. So that's what to infant and infant to adult Granger causal influences that were stronger during direct gaze. And I haven't shown in this analysis, but we did a separate analysis to show that these differences were independent of how the two brains were separately synchronizing to the speech. This is something I'm going to be talking about a bit more, and Alejandro is definitely going to be talking about. But we looked at this and we found that speech-brain entrainment wasn't driving these brain-to-brain entrainment findings that we were finding. Okay. And then in experiment 1, where we looked at the relationship between the infant and the screen, here we actually had 3 conditions. One, this last direct oblique was put in because the reviewers asked for it as well. Firstly, direct and indirect, but also with the face indirect, but the eyes looking directly at the screen. Yeah, just to make the direct and indirect conditions as close as we possibly could. So here we found relationships from the adult to the infant. Yeah, but we didn't find any relationships in the opposite direction from the infant to the adult, and that's kind of what you'd expect, because the adult is pre-recorded. So so we were kind of reassured that we were finding it in this direction, but not in the opposite direction. And again, both of these, these relationships in both the theta and the alpha band, I'm just showing adult to infant relationships here, but these relationships were weaker during indirect gaze and stronger during direct and direct oblique gaze. So we concluded from this that we'd found greater adult to infant Granger causal influences during direct compared to indirect gaze. And we also found, as a separate analysis in this paper, that infants who vocalized for longer during direct gaze also had a stronger Granger causal influence on the adult's brain activity. And that was present during direct gaze, but not during indirect gaze, this relationship. So that was a study that we did that really, personally got us started in this line of work, showing, as I say, that we get these bidirectional relationships, even in infants that are 8 months old. So those were 8-month-old infants. So one of the questions that we're going to be looking at in future is, how early do these types of things develop? We know from ERP research, so that's evoked response potential research, that infants show sensitivity to direct gaze, even very shortly after birth. So some colleagues of mine, Teresa Ferroni, did this paper a few years ago. This is just simply flashing up a picture of either someone looking directly at the screen or someone looking indirectly. And we know that babies show a different neural response to direct gaze compared to indirect gaze, even within a few hours of birth. But we don't know yet for synchrony. So that's something that we're going to be hopefully looking at in future. Okay, so that was part 1 of the talk. Part 2, we're going to be looking at what causes interpersonal neurosynchrony. So this is something that, a lot of the researchers who are working with ERP and looking at phase synchrony. Oh, sorry, I forgot to say, what I'm going to be talking about for this part of the talk is a summary of a review article that we've just published in the journal Trends in Cognitive Sciences. And here is the reference if you want to look it up more. So what I was going to say was, so one of the questions that we get asked quite often when we present this is kind of the relationships that we're looking at. So we were looking at 3 to 9 hertz. are really at quite a fine time scale. So if you think about these relationships, up to 9 oscillations a second, I know quite a few of the other researchers are also presenting relationships at a similarly fine-grained timescale. But how mechanically do two brains, influence one another at such a fine-grained timescale? It's a really, really kind of important and, quite a hard question. And it's something that we get asked a lot. So this is what we've been looking at. So we know that we get kind of alignment between the brain and temporal structures in speech. So there's a much longer history of research into how the brain aligns to temporal structures in speech over a similar types of timescale, lots looking at kind of theta. So there are some ideas that we can take from this older research tradition to look now at this new research into brain-to-brain synchrony. And one of the ideas that's quite well established in speech-brain synchrony is that the brain uses what are called kind of edges, so kind of rapid moments of acoustic transition in a speech signal. And these trigger something called phase resetting in the brain. And this process of phase resetting to the edges in speech is something that we think helps the brain to achieve and maintain speech-brain entrainment. So we're taking this same idea and saying, can we look at brain-to-brain entrainment in the same way? So what are the edges in a social interaction that might cause phase resetting, leading to us to help to establish and also maintain neural entrainment, so brain-brain entrainment during an interaction. So one of the things that we're looking at, and this is work being done by a PhD student of mine called Ira, is we're observing a free interaction between a child and their parent while we're recording brain activity in both the child and the parent at the same time. We're coding at a high temporal resolution, so 50 frames per second, the exact moments of the child's shift between the partner and the play object. So the times when the baby is looking from the partner to the play object and when the mum is looking from the partner to the play object. And so what I'm going to present you just is pilot data. So we're still collecting data for this study. So please take this with what we call in England a pinch of salt. So these results will change as we go on to collect more data. But we're basically taking the moments of mutual gaze onset. So that is the moments where the baby and the mother are looking directly at one another. And then we're breaking them down into different temporal structures. So this is looking at theta. And then we're phase sorting them. So just by the phase of the oscillation at the moment of gaze onset. So you can see these are ones that are at the trough of the phase. These are ones that are at the peak. Yeah. And then we're looking at how the phase changes in the time interval after a mutual gaze onset. So at this time, at the time of the gaze onset, we have evenly distributed phase. And we're looking at whether in the time windows after a gaze onset, you start to lead to aligned phase. And then we're calculating a measure called intertrial coherence to look at, does this change in the time windows after? And then we're doing exactly the same analysis in the baby and the parent at the same time. Yeah. So we're looking at phase resetting in the baby and the parent at the same time. And we're looking at how this affects the phase locking value, the phase synchrony. Yeah. And we're doing this to test the hypothesis that gaze onsets cause phase resetting in both sets of brains at the same time. And this is a driver of interpersonal brain-brain entrainment. Yeah? So as I say, I don't have the answers to you. We're still collecting data, but that's what we're looking at. In future, we're also planning to use similar types of analysis to look at other types of neural drivers of neural synchrony. So I've been talking about gaze as a driver of phase resetting and how that potentially might cause synchrony. In future, we're also hoping to look at speech as a driver in the same type of way, and also potentially touch. So does it drive this kind of phase resetting, leading to brain entrainment? So those are all goals for future work. So that is the first part of this talk, looking at how shared entrainment to common behavioral cues might act as a driver of interpersonal neural synchrony. But we know that this can't be the whole story. Yeah, we know that the whole reason why neural synchrony develops can't just be because of these edges during a social interaction. And we know that because some researchers have shown that even under conditions where behavioral synchrony is very tightly controlled for, we've heard, and I'm sure we will hear more about this at other moments today. These are a couple of papers. This is with children, the Reindl et al. Paper, and the Kingsbury et al. Paper is with animals. And they've shown that neural synchrony can be shown even in the absence of any behavioral synchrony. So we are also, in parallel, looking at another route to interpersonal neurosynchrony, independent of entrainment to behavioral cues. And this is the role of shared understanding. So building on some of the themes that Simone has been talking about and some of the other things that we're going to be hearing about later in the day. So what is this idea of actor-observer correspondences that I talked about? So this is an idea that's quite well established in various different fields of neuroscience, and it's actually been shown for physiology too. That when I'm watching someone else perform an action, I get overlapping patterns of neural activity between myself when I'm experiencing the action and someone else when they're watching me experience that action. So this has been shown, for example, in this paper, looking at disgust. So when I see someone else experiencing disgust, I get overlapping patterns of neural activity as when I'm experiencing disgust myself. It's also, so that's been shown with fMRI. It's also been shown in various other contexts. This is quite a well-researched phenomenon within the field of mirror neuron research. So one recent paper that I just wanted to talk to you about quickly, it's not from our group, it's from Kingsbury and colleagues in Cell, looked at the role of these types of actor-observer correspondences in driving neural synchrony. So this was done with mice, this study. And they found, even under conditions, as I say, where behavioral synchrony was very tightly controlled for, they found significant neural synchrony between their mice. But because they were recording with animals, they were able to do something that we can't do in human research, which was they actually did in vivo recording in the dorsomedial prefrontal cortex during social interactions. And they found distinct but overlapping populations of cells in the dorsomedial prefrontal cortex that coded for when I do an action myself and when I watch someone else do the same action. Yeah. So this is slightly different to mirror neurons in that they were distinct populations coding for these things, but in an overlapping part of the brain. So they were very close to one another. And they found that when they removed these cells from their calculations, the interpersonal neural synchrony that they had been observing disappeared. So they concluded from this that these kind of neurons that show similar patterns of neural activity between watching someone else perform an action and performing it oneself, they said that these were kind of driving the interpersonal neural synchrony they observed. So this is, we think, interesting kind of research. Obviously, because it's done with animals, we can't do exactly the same thing with humans. But we've done some research that we think maybe is a little bit similar to this. I'll let you judge for yourself whether it is or not. So this is two papers, one that was published in PLOS Biology in 2018, and one which is under review at the moment. I'm just about to post it on bioRxiv. And what we did was, again, for this research, we videoed babies and adults during a free play interaction. And again, we coded at a high time resolution, so to the nearest frame, the moments of the child shifting gaze between play objects and from the play object to the partner. Okay? So first of all, we looked at the relationship of the baby's brain activity to the baby's attention. Yeah. And what we did was we coded the baby's attention to the object just as a series of ones and noughts. So there was just one object on the table, and we coded a 1 if they were paying attention to the object and a 0 if they were distracted and not looking at the object. Yeah. And then we took out infant brain activity to start with in the 1 to 2 hertz range. And then we simply calculated the cross correlation across the whole time series between the brain activity and the looking behavior. We did the same thing for all of the participants and averaged it. And then we drew that as the first row on this plot that I'm going to be building up on. Yeah. So it's blue, suggesting that there was a low cross correlation overall. And then we did the same thing looking at brain activity in the 2 to 3 hertz range. And again, we correlated it with a looking duration. And then we calculated the cross correlation, and we did the same thing for all of our participants. And then we drew that as the next line. So this is showing EEG in the 2 to 3 hertz range. And then we repeated it for all of the different frequencies of brain activity. And when you repeat it, you get a plot that looks like this overall. So this suggests that when I look at the relationship between infants' brain activity and their own attention, you get a strong association in the theta-ish range, 5 to 8 hertz, but you don't get associations between brain activity and attention in other frequency ranges. So we can use this analysis to look at the relationship between infant attention and infant brain activity. We can also use the same analysis to look at the relationship between parent attention and parent brain activity. But we can also look across the dyad. Yeah. So we can use exactly the same analysis to look at the relationship between infant attention and parent brain activity. And we can look at the other way between infant brain activity and parent attention. Yeah? So when we look at infant attention to infant brain activity, we get similar to what I showed you. This is a slightly different electrode. So this is looking at anterior. So this was AFZ, so here at the middle. And this is posterior. So this was POZ at the back. And we get these associations, which are stronger in the anterior compared to the posterior. When we look at the same relationship between the parent's attention and the parent's brain activity, we get an association at a slightly higher frequency range that again is stronger anterior than posterior. But the really interesting thing is when we look at the relationship between the infant attention and the parent's brain activity. Yeah. So here we get an association between the parent's brain activity and the infant. And again, I'm not showing you this, but in separate plots, we did another analysis to show that this isn't just to do with the fact that the parent's attention follows the child's attention. Yeah. This is, we think, a direct relationship of the parent's attention tracking the infant, the parent's brain activity tracking the infant's attentional fluctuations. And then when we look in so this is, yeah, as I just said. And then when we look at the opposite direction, so this is the infant's brain activity tracking the parent's attention, we get the same thing, but a weaker relationship. So this is significant. I'm not showing you the significance plots, but it is a significant relationship. But it's a weaker relationship than it is in the other direction. So we think that these results show that both partners' brain activity is tracking the attention partners of the other, independent, as I say, of their own attention pattern. And we have results that all of these relationships, including the tracking, are stronger at these electrodes than these electrodes. It's very hard to get more temporal resolution than that with EEG. And we think that maybe, although I'll let you judge for yourself, that these types of tracking, I'm tracking what my partner is attending to, maybe they are similar to these actor-observer relationships shown by Kingsbury. So maybe they act as drivers of interpersonal synchrony, even though in these analyses we haven't looked directly at brain-to-brain synchrony. We've looked at the relationships between behavior and brain activity across the dyad. So, just to say what I said, so in part one, I was presenting a study showing that aspects of interpersonal neurosynchrony develop by 8 months of age. And then I've talked about this idea that we talked about more in this review that I mentioned, asking the question, what causes interpersonal neural synchrony? And we talked about two quite different potential causes of interpersonal synchrony. One, shared entrainment to common behavioral cues, and other to actor-observer correspondences. And then finally, just to lead into Alejandro's talk, which is coming next, we're also interested, and again, building on a lot of the great research that Simone was talking about earlier, about the role of shared understanding in driving synchrony. Yeah. So we know from some other papers with adults and infants that interpersonal synchrony is very important for understanding and learning. And I think that next you're going to be hearing about some studies showing that adults show brain-to-brain entrainment during speaker-listener tasks beyond that which is expected based simply on speech-brain entrainment. So in future, we're hoping to look at this question for the perspective of early learning. Yeah. So how does a child's early learning and early understanding, what is the role of understanding in driving neural synchrony during early learning interactions? Just very quickly before I finish, we've got some opportunities for postdocs and PhDs starting in the lab who are going to be looking directly at these questions. So one starting soon, another in 2021-22. So please do get in contact with me if you've got any questions. My email address is up here. If you've got any questions and you want to get in contact, do ask, or on Twitter. But for now, thank you very much to my colleagues who helped with this, to the funders, and to our research participants.
Okay, Dr. Ross, there is a question in the chat window. So, what do you think the role of parent-infant interaction or attachment is in the ontogeny of interpersonal synchrony? Moreover, is there evidence suggesting that interpersonal synchrony propels learning in infants?
Okay, so that's kind of two questions, and they're both very good. Okay, so that's kind of two questions, and they're both very good questions. About the role of attachment, I'll leave you to the real experts on that, who's Pascal, who is talking later on that. I know that Pascal has done some research that he's going to be talking about, looking at with FNIRS at entrainment and some associations between that and entrainment. Yeah, we've also done some stuff on physiology that I haven't talked about today to look at shared entrainment during parent-child entrainment in physiology. If anyone's interested in finding more about the role of attachment in physiology, email me, and there's lots of interesting papers that I can look at. I can send you for that. In terms of the role of learning, the short answer is we don't know at the moment. We've got a paper that is under review at second round review, looking at associations between phase entrainment in adults and children and that relationships to learning. I know that some other research groups have got some papers on this. Just quickly, one project that we're doing, we're just setting up at the moment, is looking specifically at the role of so almost all of the research and learning is from the point of view of the person who is teaching, communicating to the person being taught. Yeah. So it's looking at the one-way flow of information from the teacher to the learner. Yeah. And what we're trying to do is there's some behavioral research suggesting that even when a young baby points at something, yeah, if you teach them something after they've pointed at it, so if you teach them the label for an object after they've pointed at it, they remember that label better. Yeah. So what we're looking at in future is looking at the role of babies as active participants. So when they shift to a new object, does that change the bidirectional dynamics of the relationship? Yeah. So it's not just looking at how well I, as the child, can entrain to the neural oscillations of the person who is teaching me, but more as a bidirectional relationship. Yeah. But the short answer is we don't have any findings on that yet. But in a few months, if you're interested, email me and I can send you the paper when it's available.
Thank you. The next question is, mother and father play different roles in child-parents interaction. So did you investigate this point in your study?
So that's another really good question. So that's another really good question. The short answer is no. And it's really boring that they have to come to the lab, and it's hard to, and we have to do it during the office hours. And it's easier to find enough mothers to come in because more mothers take time off still. About 80% of our parents are mothers and 20% are fathers. But it means it's very hard to get enough power to look at fathers. So we haven't looked at it from the neural synchrony work. It's a really, really interesting relationship. Again, there is some interesting research looking at physiological synchrony. So how that differs between mothers and babies and fathers and babies. So again, if you're interested, email me and I can send you those papers. But the short answer is it's a really important question that we just haven't been able to look at yet.
Oh, okay.